So we are beginning our today's session of the Science Dialogue series, which is basically on gene therapy bench to bedside in decade, presented by our today's speaker, who is Dr. Sanal MG. Hand over this virtual platform to you, so that without any delay we can start your presentation.
Good evening, everybody. So today I'll be talking something close to my heart. It is gene therapy. I'll be actually reviewing uh, the topic briefly, a brief history, and the recent developments. So, what is gene therapy? It's an experimental technique that involves modifying or manipulating genes within a person's cells to treat or prevent disease or change a genetic trait. This can be done in several ways. You can replace a mutated gene, which is uh, causing the disease, or inactivating a mutated gene, or by introducing a totally new gene. So a gene is essentially the storage of genetic information. From that perspective, you don't have to code a protein. So that means this gene therapy also involves non-coding genes. So this is the outline of my presentation. I will start uh, with classic gene therapy. Then briefly I will cover gene editing and some of the clinical trials, old and new. And we will also discuss gene editing in specific diseases and lab grown models and efficiency with respect to gene therapy and a conclusion. The classic gene therapy, the concept was conceived way back in 1960s, and in 1972 by Theodore Friedman and Richard Roblin, who proposed gene therapy in a paper published in Science, opening with gene therapy may ameliorate human genetic disease in future. The introduction of retrieval system marked a significant milestone in gene therapy and molecular biology because it paved new avenues into the field. Before that, delivering a gene was so difficult and inefficient. Gene therapy you can do in multiple ways. You can classify it in multiple ways. One way to classify is the gene therapies as in vivo and ex vivo. So in vivo gene therapy, it is very simple. You deliver the gene intravenously, directly into the patient. The gene can be put inside a plasmid or a viral vector like lentivirus or adeno-associated virus or a virus of your interest, or you can use lipid nanoparticles or any suitable carrier. Ex vivo gene therapy, on the other hand, is done in a petri dish. You extract cells from the patient, then you can transduce them if you're using a viral vector or transfect them using lipid nanoparticles or by electroporation, or there are several methods. And these transduced cells from the patients are now put back or infused back into the patient. So essentially, you are taking cells from the patient, replacing the gene or altering the gene or putting a new gene, totally new gene, and the changed cells are selected and put back into the patient. That is ex vivo gene therapy. As I mentioned you, there are several ways to deliver genes or gene editors. Classic viral vectors. You can use retrovirus, lentivirus, adeno-associated virus, adenovirus, fomavirus, sendai virus. There are a lot of viral vectors. And the non-viral methods include, as many of you know, liposomes, nanoparticles, electroporation, microinjection, and fusogens. And fusogens are generally proteins or non-protein molecules which induce fusion of a cargo with a cell. You can also use exosomes, for example. Now, I will go into gene editing. Gene editors are tools and technologies used to modify DNA of an organism. They allow precise changes to the genetic code, which can be used for a variety of purposes, including research, medicine, agriculture. And commonly used gene editing tools are zinc finger nucleases, TALENs, CRISPR-Cas, bridge RNAs, meganucleases. All these are examples for gene editing tools. However, because of the ease of use, the ease of design, CRISPR-Cas is more used. Plus, there is it plus another reason for using CRISPR-Cas is that zinc finger nucleases have several patents, several restrictions. That many restrictions are not there in the use of CRISPR-Cas. Bridge RNAs are an evolving technology, which which I will talk later. So there are several factors which affect the efficiency of gene delivery or gene therapy. One thing is that the gene delivery itself is quite inefficient because if you're using a virus, the infectivity of the virus may not be poor. Even the one virus may infect dividing cell, other virus may not infect dividing cell. Or some virus may have an affinity to a peculiar tissue type, while some other virus are non-specific infectors. Similarly, the same is true with nanoparticles also. Certain nanoparticles are highly specific for an organ or tissue, but while others are not. In fact, you can engineer the specificity of nanoparticles and liposomes used for gene delivery. Another problem which is usually faced in gene therapy is the immune reactions once you infuse a viral vector or a non-viral vector. This is more common with viral vectors. So the body may produce antibodies, or there may be existing antibodies which will completely neutralize the virus. So there are no sufficient copies of the virus left in blood to infect your target organ. So the gene therapy fails. So now I'll go to CRISPR-Cas system. All of you know, perhaps I don't have to explain you much. There are so many lectures in YouTube or reviews you can find in PubMed. So I will not go into the details, but as most of you know, the system involves developed as an immune system as a bacterial immune system against invading pathogens, especially virus or invading hazardous plasmids. So what the bacteria does is it captures a piece of or certain pieces of the genetic elements of the invader, the invading virus, for example, and makes it its own. That means the invaders genes, a parts of those genes become the part of the host, part of the bacteria. And these elements are used to recognize the invader, the invading virus. So as you can see here, the invaders gene sequences are crushed into pieces and assimilated into the genome, and which is later transcribed into an RNA. That RNA that is used to recognize the invading pathogen. And now with the help of a protein called Cas9 protein or its sisters and brothers, the invader's genome is cleaved into multiple pieces or the invader is neutralized. So the canonical CRISPR-Cas system has a PAM sequence which is associated with, for example, SP-Cas9. And different PAMs have been found to be associated with the bacterial Cas9 of Streptococcus thermophilus, which is called ST1 Cas9. Neisseria meningitidis, it will be NM-Cas9. And so Treponema, for example, it will be TD-Cas9. Francisella, it will be FN-Cas9. So each of these Cas9 systems have its own properties. These Cas9 orthologs are and their engineered variants recognize different palm sites at any desired genome location for precise editing with altered and improved palm specificities. NCAS or Cas9 nickase is a modified version of the Cas9 protein that can cleave single strands of DNA. Cas12a, which is derived from Pivotella and Francisella, these are two types of two different bacteria, needs only sgRNA. It does not require the tracer RNA and provides a T-rich PAM recognition site for genome editing and generates sticky ends that are more effective for precise DNA insertion. CRISPR-Cas13 from bacterium Leptotrichica trichia shahi is characterized as a RNA-guided CRISPR system that targets RNA but not DNA sequence. That means this can be used very well to cleave RNA or dictate RNA. So that means it can be used against RNA virus, or it can also be used to dictate RNA viruses, for example, COVID or hepatitis C virus, which are RNA virus. That means it can be used as a diagnostic tool. Now I'll briefly speak about base and prime editing. How base editing works. So there will be a guide RNA. Guide RNA guides essentially the Cas9 protein to the target. So it directs the base editor to the specific location of the genome where the editing is to be made. So what happens? Here, the Cas9 is essentially a DNA cleaving element. It's a DNA chopper. Okay, it cuts DNA into multiple pieces. So this Cas9 protein is guided by what? The gRNA, the guide RNA. So the guide RNA recognizes the element DNA which needs to be cut into pieces. So here the basic, so that is how the guide RNA works. Here, what is done is that when you do, that is how the classic CRISPR-Cas works. Here, what is done is that the Cas9 protein is engineered. So how it is engineered? The enzyme is with an enzyme which removes or deaminates, removes the amino group from a nucleotide. For example, as you know, adenine has an amino group. When you remove the amino group, what you will get? A hypoxanthine, right? So guanine is deaminated and you will get xanthine. So here,
प्रेजेंटेशन बंद हो गया क्या?
hello.
चल तो रहा है. It's moving. Guide RNA, he's explaining.
Yeah. So good that you are hearing. So in essentially what happens is that cytosine, for example, can be deaminated to uridine, and uridine is going to pair with thymine. to in essence, what you can do is that you can change one nucleate to another without actually cutting the DNA, which makes the entire process much safe. So similarly, adenine will be deaminated to inosine, and during the replication of the DNA, it will be that peculiar strand will, due to base pairing, will be converted to guanine. So essentially, you can convert adenine to guanine the same way as I told about cytosine. The cytosine will be deaminated to uridine, and when the replication is happening, then finally on the other strand it will be replaced by thymine, as I was telling you. So this is what the advantage of this system. You really don't have to cut the DNA. If you cut the DNA, you end up with mutations, which all of those mutations are not very predictable. Now comes the glycosylase base editors. Here we use engineered. Uh, this is a bit more complicated. So here glycosylases are used instead of deaminases. So what happens here is that, as you know, in DNA, there is a sugar which is attached to the nucleate. So this sugar is chopped off. That means you are essentially cutting the backbone of the DNA. When that happens, the next thing what happens is, when a mutation happens, a DNA is chopped inside a cell, what happens is an immediate reaction. This will lead to the base excision repair pathway. This is the natural mechanism because DNA damage is an emergency in any cell. So immediately the base excision pathway comes into action. So what follows is a natural repair. So during that natural repair, what happens is that the damage, so we will always provide an intact DNA, or a repair template, and that template will be replaced. Will am I audible?
Yes,
Hello?
sir. Yes, sir.
Yeah. So what are the features of this?
Dr. Sanal,
The
sir,
glycosylase base editors can target bases that are difficult to edit with traditional base editors such as guanine and thymine. They
sir,
have high specificity.
Sanal,
Yes,
sir.
please.
Sorry to interrupt. Actually, there are two persons who are asking the same question. What is sgRNA, small guide RNA?
Yeah. So these are engineered. So as I have, or shall I address all the questions at the end, or?
Uh, Actually, just
Okay,
as
okay, it's fine. I'll tell you. So uh, you can see here, there's a tracer RNA. Can you see a tracer RNA? So
yes,
can
sir.
you see a tracer
Yes, sir.
RNA? So in this slide, you can see a tracer RNA. This tracer RNA is very much required for the maturation of the pre-Sierra to of the pre-CRRNA to the CRRNA. So what are the difference between this? The pre-CRRNA, as you can see, is a long strand which undergoes maturation. What happens during maturation is very specific cleavages. For this specific cleavages to happen, the tracer RNA is required. So now what all you have here? You have a tracer RNA and a pre-CRRNA, and which will give rise to a CRRNA. So essentially, you need two RNA elements for the CRISPR-Cas to work. One is a CRRNA and a tracer RNA. So will it be intelligent to fuse this together and make it one? So it will be very easy for us to express in cells. So you are going to combine tracer RNA, the violet one, and the cRNA. So that synthetic product is called sgRNA or single guide RNA. Because two things are fusing together to make a single element. So it is called a single guide RNA. Hope I answered your question.
Yes, sir.
Okay. Now there is something called prime editing. So essentially, this CRISPR-Cas system is like molecular scissors, which are cutting the DNA. Once the DNA is cut, the body's own system is prepared for a DNA repair. If you had to repair a gene or a mutation, then you should have the right DNA, right? You had to provide the right template for the repair. So usually what we do is that you transfect or transduct along with the CRISPR-Cas system, the template DNA also. Here in prime editing, what they have done is quite interesting. Here, as you can see in the picture, there is a guide sequence. See the 5' end? You can find a guide sequence in blue color, and you will find a hairpin and a pre. Can you see in the brown color? Okay, the peg RNA, or it's a so then it is, which is after that this system is fused with a reverse transcript. Then this Cas9 in light brown is fused with a reverse transcriptase. You will get a fusion protein, which is a Cas9 reverse transcriptase fusion protein. So, and when you see the RNA sequence, you can see here uh, the guide RNA is followed by a reverse transcription template with the corresponding edit, which is the brown element, which is between the yellow elements. Can you see a base change there? That is the edit. So that is the nucleotide which you are going to replace. Okay, so what is going to happen is that the reverse transcriptase, there will be a reverse transcription of this RNA into the DNA. That will generate a DNA template for the homologous recombination. That is how the prime editing works. The, guided by the PEG RNA, NCAS produces a single strand break at the palm containing strand to expose the 3' flap that hybridizes with the PBS of or the primer binding site of the PEG RNA, and the 3' flap will be extended by the reverse transcriptase using reverse template containing the Bedside edit. Now I will come to the clinical trials and FDA approved therapies. This is the first ever successful gene therapy. It was done by William French Anderson, who is known as the father of gene therapy. You can see him in this picture. Anderson, who is on the left side, and you can see two girls who underwent the first gene therapy. The first ever gene therapy was done for adenosine deaminase using a retroviral vector. Her name was Ashanti De Silva, the first patient to undergo gene therapy. She received multiple infusions of genetically modified cells, and her immune system was improved significantly. As you all know, adenosine deaminase deficiency is also called as bubble baby syndrome. Why? Because those kids who suffer from adenosine deaminase deficiency, their immunity is very poor. They are highly susceptible to recurrent infections and they die of infections. So they have to be kept away from germs all the time. So they had to be extremely careful when it comes to hygiene. So essentially what is done is that these kids will be kept in a bubble or a very or a highly clean room. That's why they were called bubble babies, because if they go out of these clean rooms, they will get immediately infection and they will die. So this girl was saved by using adenosine deaminase gene therapy using a retroviral vector. The second patient, Cynthia, also experienced a significant improvements in her immune function after the gene therapy. So the patients are happy. How and Ashanti is doing very well. As of October 2024, Ashanti De Silva will be 38 years old now. Otherwise, she would now. It is very unlikely that she would have survived her childhood. However, the doctor is not doing well. Unfortunately, he was jailed for a couple of years for molesting his assistant's young daughter for years. And now let me come to the bad side of gene therapy. There was a terrible death from gene therapy. Jesse Gelsinger, 18-year-old, in 1999, died of a gene therapy trial, which happened in University of Pennsylvania. Gelsinger suffered from ornithine transcarbamylase deficiency, an X-linked genetic disease of the liver, which causes hyperammonemia. On September 13th, 1999, Jesse was injected with an adenoviral vector carrying the corrected gene to test the safety of the procedure. He died 4 days later, apparently having suffered a massive immune response triggered by the virus. That is the problem which I told you. When you get infected with a virus, the body responds very strongly. And we use the very virus for gene therapy. So when you infuse this virus containing the corrected gene, sometimes your body responds with a massive immune response, which can cause sometimes an unfortunate death. There's also another problem, especially with integrating virus. As I told you before, there are different types of viral vectors. Lentivirus, which are derived originally from HIV, are integrating virus. That means they integrate with your gene. That is good. Yes, that is good, because once you are infected with that virus, which carries the gene of interest or the corrected gene, that gene stays with you for your life. That is good. What is bad? Because you cannot always predict where the gene is going to integrate. Sometimes the gene may integrate at a wrong place. So if the integration is near a proto-oncogene, probably the lentiviral integration may trigger the oncogene. Exactly this happened in few cases. The first case of leukemia linked to a gene therapy was reported in 2002-2003 in the French SCID trials. SCID means severe combined immunodeficiency, the same disease which I was describing you. Severe combined immunodeficiency. The cause was a retrovirus integrated near the LMO2 oncogene, leading to its activation and development of leukemia. So these cases prompted a re-evaluation of gene therapy safety and led to the development of safer vector. So I am telling about an old time, 2002, 2003. So at that time, the first reports of cancer came because of aberrant integration of lentivirus. After this, scientists started developing self-inactivating vectors, which have a lower risk of causing insertional mutagenesis. This is the big problem with lentiviral vectors. Another way is to totally avoid integrating vectors like lentivirus or retrovirus. Simply use adeno-associated virus. Adeno-associated virus do not integrate. They stay inside the cell like an episome. But what is the problem then? The problem is that as cells divide, the, the number of the copy numbers gets diluted. That means after a couple of divisions, cell will not contain any virus. So the gene therapy fails there. It will work for some time, but then fails. So that is a problem with adeno-associated virus, especially in those cells which are fast dividing. However, there are tissues which don't divide or rarely divide. For example, your brain. So adeno-associated virus is very suitable for brain gene therapy, or is also useful for liver gene therapy in certain diseases where the rate of cell division is less. Although liver regenerates very fast, in a normal liver, the cell division is less frequent compared to bone marrow or intestine. So what is the current status? Gene therapy and bone marrow transplant for multiple sulfatase deficiencies. What are sulfatase deficiency? Probably you might have heard the story of Bechavat. Bechavat was a Bengali scientist who first discovered the cause of uh, a severe neurologically debilitating disease called metachromatic leukodystrophy, which leads to death of the those patients. So to alleviate the symptoms, or you can use you can now use uh, gene therapy. Preclinical studies in mice have shown were promising. However, it was in that case it was just a bone marrow transplantation. But in there are other uses for gene therapy. For example, it can be used for extensive stage small cell lung cancer to assess. So small cell lung cancer, as perhaps you know, is a type of lung cancer which is very difficult to manage. So some of the gene therapy trials are partially successful in extending the life in small cell lung cancer. And Beam Therapeutics launch of alpha-1 antitrypsin deficiency treatment was also quite successful. SERPINA1 is the gene which is defective in alpha-1 antitrypsin deficiency. Alpha-1 antitrypsin deficiency is a disease which affects lungs and liver mainly. This is because of the deficiency of an important enzyme called alpha-1 antitrypsin, which fights against the proteolytic activity of trypsin, which is a protease. So if proteases are active, very active, when secreted, they can simply digest the cells. Leukocytes secrete, as you know, in your stomach also, trypsin is secreted. So many cells in our body, many tissues in our body, there are leukocytes because they are everywhere. They secrete elastase, trypsin, and several other proteases. And these proteases, if they go uncontrolled, can chew the very cells. So it can damage several organs. So lungs and liver are severely damaged in alpha-1 antitrypsin deficiency. So In a clinical trial, they they used gene therapy, which is using base editing, which I discussed some time back, to treat alpha 1 antitrypsin deficiency. And Intellia Therapeutics launched a gene therapy for hereditary angioedema. It's another disease where people get uh, a lot of inflammation, means their face or their body just swells up without no clear apparent reason. A small trigger, with a small trigger, sometimes their face swells out, their lips may swell out, and this is a big problem. It can also can cause sometimes death because of suffocation, for example. So a gene therapy was launched, and I'll talk about that later. And another disease which was amenable to gene therapy is hemophilia A and hemophilia B. To treat hemophilia A by delivering a healthy copy of factor VIII gene to liver cells using a modified adeno-associated vector. So this was a metagenomics trial for hemophilia A. So Racorsa, in combination with pembrolizumab in previously treated lung small cell cancer. This is what I was telling you. These are the results of Acclaim 1 and Acclaim 3 trials, which uh, gave some progression-free survival and partial remission for the patients who underwent the combined therapy. This acts through TSC2, which acts as a tumor suppressant. It promotes apoptosis in cancer cells. So the gene was encapsulated in a non-viral nanoparticles made from lipid-based molecules. And this was used in combination with the pembrolizumab, which is an immune checkpoint inhibitor. And there was a hemophilia A gene therapy using adeno-associated virus 5. So here you should know that adeno-associated virus has several genotypes, type 1, type 2, type 3, like that. So in this peculiar gene therapy trial, adeno-associated virus 5 was used. And the treatment was given to about 134 patients. And the mean factor 8 level increased following the treatment. 80% of those treated became almost bleeding free. This is a great achievement. However, there were some problems. So AV5 was used, not which is not a very liver specific. So as I was telling you, certain virus serotypes have a higher affinity for certain organs. For example, AV3 has a huge affinity for liver. So instead of using AV3, they used AV5, which means the affinity of the virus to liver is low. So to compensate for that, they had to use a very high dose of 600 to 10 to the power of 13 copies, and this can cause liver inflammation. So serious adverse events was found in 16% of the patient who underwent treatment. And now, this year, we saw the first successful gene therapy trial from India. It was carried out at CMC Vellore. Congrats to Alok Srivastava and his team. So, what they have done is that from the circulating blood, they isolated circulating CD34 positive hematopoietic stem cells. These hematopoietic stem cells can give rise to any cells in that lineage, means neutrophils, macrophages, lymphocytes, all those things. So they isolate the CD34 positive stem cells and they transduce it with a a lentivirus containing factor 8 because factor 8 deficiency causes hemophilia A. And what happens to if this hemophilia patients are not treated? These patients suffer from severe bleeding, especially bleeding of joints. It makes the life a hell for, and so that means even with a minor fall, they can get their knees or other joints can become so swollen because blood gets into the joint. Repeated bleeding causes destruction of the joint. After some time, they will not be able to use that joint, for example, the knee joint. So patients cannot walk properly, plus immense pain and suffering. This is the problem of hemophilia A. So with gene therapy, now they are practically, most of the people whom they have tried were cured of hemophilia. But what was the problem with this trial? The first problem was that you have to myeloablate the patient. Why? Because you are collecting the CD34 positive stem cell from the patient's blood, and these isolated cells are grown in culture. Then you are using a lentivirus to transduce them with a correct copy of factor 8. Now you have a nice copy and your disease is corrected. But the problem is that if you put these cells back, only a very, very, very insignificant portion of your bone marrow cells are corrected because you are not going to get several millions of cells from circulating blood. Even if you take the isolate from bone marrow, you may get more hematopoietic stem cells, but still, compared to the volume of the total bone marrow, this is minuscule. So what is usually done is that the patient's existing bone marrow is ablated or killed. That means these patients undergo a total bone marrow depletion. This was originally done through by exposing the patients to severe radiation, X-ray radiation or gamma radiation. Now there are better methods which are less damaging. So you can use certain drugs like busulfan, for example. So such an approach was used in this trial to myeloablate the bone marrow. So all the bone marrow cells of that patient are killed, and after killing all the bone marrow cells, those gene edited cells, or sorry, not gene edited, but transduced hematopoietic stems are infused back. And since they are stem cells, they can divide a lot and repopulate the entire bone marrow within a couple of days or weeks. But couple of days or weeks is not a small time. In this time, these patients can get severe infections because if they are not careful, they can get severe infections because there is no bone marrow. This means there are no your there are no sufficient number of white blood cells to fight infections. So these people who are bone marrow ablated are highly susceptible to severe infections. So all these patients has to undergo through a window period where they can die because of severe infections. That is one problem with this trial. And another aspect is that they have used, as you can see in this paper, they have used a CD68 promoter. CD68 promoter, so the factor 8 is expressed under CD68 promoter. So CD68, as you know, are macrophage, cells of macrophage lineage. So that means, and macrophages, as you know, or most of these leukocytes are highly responsible, are respond to infections. That means their number goes up when there is an infection in the body. This also means that when these patients who have undergone gene therapy are exposed to some sort of infection, the CD68 positive cells, which are the macrophages, will divide very fast. You will have more macrophages, not macrophages will divide very fast. Other than that, you will have more macrophages secreting a lot of factor VIII. Will this cause clotting problems? We do not know. Plus the risk of integration because they have used the lentivirus. Despite all these shortcomings, we should be proud of this trial. Now, you can use not only use gene therapy for cancer or hereditary diseases, but you can use gene therapy for infectious disease as well. A good example is AIDS, which is caused by, as you all know, HIV or human immunodeficiency virus. So there is a famous patient from Berlin. He was called as the Berlin patient who had HIV AIDS. This patient suffered from okay. One problem with HIV infection is, as you know, they are susceptible to for infections. They so they can get infected with various viruses, and some of the viruses can cause cancer also, especially leukemias. So anyway, somehow this patient got leukemia. This HIV patient got leukemia. And for leukemia, one of the treatment option is bone marrow transplantation. Therefore, this patient was underwent a bone marrow transplant. And quite surprisingly, not only he recovered from leukemia or blood cancer, but he also recovered from AIDS. That means two birds in one shot, and people were wondering how this happened. Then, following investigation revealed that HIV virus requires a special receptor to infect white blood cells, so CD4 blood cells. And this receptor is called CCR5. It is one of the receptors which are very critical in HIV infection. So, however, HIV cannot bind to a mutated receptor. That mutation is called CCR5 delta 32. If you have this mutation or some other mutation, certain other mutations are also there, HIV virus will not be able to infect you. So this opened a possibility that if you mutate the stem cells, the bone marrow stem cells of these patients for the CCR5 receptor, you can make HIV patients or common man immune to HIV. So this is the basis of development of gene therapy for HIV. It can be done in multiple ways. One way is to use a lentivirus expressing short hairpin RNA against CCR5. So again, you know perhaps what is RNA interference. So RNA interference causes there are certain RNAs which are complementary to the messenger RNA. They will go and bind to the messenger RNA, and this binding triggers a set of enzymes which will result in the chopping of the messenger RNA. That means if there is no mRNA, there is no protein. That means if you can chew away the mRNA of CCR5 protein will not be expressed, and if CCR5 protein is not expressed, you are immune to HIV infection. And another approach is through gene editing. So gene editing, as I told you, you can do gene editing using different methods. One of the popular is CRISPR-Cas9, and even before CRISPR-Cas9, people used to edit uh, DNA using zinc finger nucleases and talents. And that's a very big topic which I can cover perhaps another time. Now there are over 100 CRISPR clinical trials ongoing. We will have a peek into the FDA approved gene therapies in clinical trials. LifeGenia is an FDA approved lentiviral gene therapy designed to treat sickle cell disease in individuals aged 12 years or older who have a history of vaso-occlusive events. Elividis is a recombinant adeno-associated virus delivered gene therapy for Duchenne muscular dystrophy. Yeah. So I told you that adeno-associated virus is a good vector because it does not integrate, and it can be used for those tissues which are non-dividing. As far as muscles are concerned, muscles are essentially non-dividing cells. They don't divide that frequently. Okay, of course they go undergo hypertrophy when you exercise, but division, they do divide also, but division is not very frequent. So AAV can very well be used to treat Duchenne's muscular dystrophy. Perhaps all of you know what is Duchenne's muscular dystrophy. And what is interesting about dystrophin is that it is one of the biggest gene in human genome. And what is the biggest problem of what is one of the shortcoming of adeno-associated virus is that its cargo capacity is very small. What do you mean by cargo capacity? The ability to carry a cargo. For any viral vector, what is the cargo? The gene it is carrying. So it cannot carry the dystrophin gene because it is too big for adeno-associated virus. Because adeno-associated virus is a small virus and it can carry genes up to 4 kilobases. Not more than that. Even if it got, because larger the gene, the packaging of the virus becomes inefficient. So what they did is very interesting. They made a shortened version of dystrophy, which is only 138 kilodalton compared to 429 kilodalton dystrophin protein. And this was infused intravenously. So another marvelous achievement was Kebilidi. Kebilidi is a treatment for patients who are suffering from aromatic L-amino acid decarboxylase. I will tell you what this disease is. It is mainly affects the brain and nervous system. And Kebilidi is a adeno-associated virus-based gene therapy. It is also delivered by intravenous infusion. Now comes Len-Medley. Len-Medley is used for metachromatic leukodystrophy. It's a CNS disease, which I have already discussed with you. So here, brain is not targeted, but instead bone marrow was targeted because bone marrow, even if you target bone marrow and bone marrow derived blood cells secrete aryl sulfatase, at least that will be sufficient to some extent to ameliorate the intensity of metachromatic leukodystrophy. The same, I told you about this disease before in the starting of this lecture. This is a gene. The who discovered aryl sulfatase? An Indian scientist, a Bengali scientist called Bechawat. So this is what happens to patients who suffer from aromatic L-amino acid decarboxylase deficiency. Babies with severe aromatic amino acid decarboxylase deficiency usually present to the clinicians during the first few months of life. Symptoms can include hypotonia, that means you will become floppy, or it's called floppy babies. These babies are floppy babies. Developmental delay, and your eyes will not move as expected. That is called oculogyric crisis, and you will have poor balance. Then difficulty in initiating and controlling movements, and gastrointestinal motility means your food will not go to the food pipe and intestine properly, because that also require nerves. The muscle contraction, the intestine or stomach requires the action of nerves. So these children also suffer from autonomic dysfunction, because that's again the autonomic system of nervous system, and difficulty in controlling temperature, blood sugar, excessive sweating, nasal congestion, and all those things. So this disease was also treated or was amenable to gene therapy, and an AAV-based gene therapy was designed, and this trial was quite successful. And one of the biggest success in gene therapy is the treatment for Leber's congenital amaurosis. This is a disease which causes blindness. This was again an AAV2 based trial, AAV2 based gene therapy. The virus containing the corrected gene was injected into the subretinal space. Again, why AAV was used? Because the affected tissue is retina, and retinal cells don't divide. They usually shed, but they don't divide. Only the tips are shed. So retina is a good tissue for adeno-associated virus-based gene therapy. And the gene involved is actually uh, quite important in the proper function of the photoreceptor cells. You know that retinoids are important in photoreception. So this enzymes is a retinoid isomerase, isomerohydrolase enzyme. Without this enzyme, the photoreceptors will not function properly. So the first gene therapy used and which was successful, and it was marketed as Luxturna. It was one of the most costly gene therapy. Now I will go into some gene editing therapeutics. These are the CRISPR clinical trials. 42% roughly are cancer trials, gene therapy trials. Then 2% are skeletal, and 5% visual system. About another 5% is immune system, and 8% endocrine system, and respiratory system like cystic fibrosis falls about 10%, and hematopoietic stem cells and hematopoietic system is another big area of gene therapy, about 21%. So about 65% of the gene therapy goes into non-genetic diseases and 35% for genetic diseases. Zinc finger based gene editing.
Sanal sir,
Yes.
sorry to interrupt, sir. How many slides are remaining?
So probably a couple of slides. I'll wind up soon.
Okay, okay, sir.
So zinc fingers, as I told you, perhaps know about zinc fingers. Zinc fingers are transcription factors. So transcription factors have a unique ability to bind to specific parts of the DNA. This very property is used for engineering zinc finger based uh, gene editors. So zinc fingers there are different transcription factors, and different transcription factors have affinity to different DNA regions. So this means that you can engineer the zinc finger proteins in a modular fashion to generate several permutations and combinations to address practically any segment of DNA. Zendiglo was approved in 2022 for transfusion-dependent beta thalassemia. And SB525 is a gene therapy for hemophilia A. Then recently, Casgevy was approved for transfusion-dependent beta thalassemia in January 2024. So this results in increase in fetal hemoglobin levels, reducing or eliminating the need for further blood transfusions. How it acts is quite interesting. Here in thalassemia, as you all know, there is the hemoglobin is affected. You know, the hemoglobin has alpha and beta chains. So, but the ratio between alpha and beta chains are disproportionate in thalassemias. So here, what they have done is a clever way. Human beings have different types of hemoglobins. When we are inside the womb, we have something called a fetal hemoglobin, which becomes adult hemoglobin. So fetal hemoglobin is suppressed when we become adults. So what they have cleverly done is to bring back the fetal hemoglobin in those people who are suffering from beta thalassemia. This means instead of a hemoglobin A, they will start producing hemoglobin F, which is fetal hemoglobin. This is achieved through by a protein called BCL11A, which is an erythroid enhancer. So this BCL11A actually suppress the production of fetal hemoglobin when somebody becomes an adult. So what they have done is that they found a clever way to target BCL11 gene, which normally suppresses the fetal hemoglobin. So gene editing in cancer, there are three types of cancer gene therapy. One is chimeric antigen receptor, which you all are familiar, and less head is TCR T cells or T cell receptor engineered cells. And then comes tumor infiltrating lymphocytes. So I'm not going into the details of CAR T cell therapies because it's a big topic. And there are key differences between CAR T cell therapies and CAR and TCR T cells. CAR T cells does not use MHCs to recognize the protein which they are targeted against. Right. On the other hand, TCR cells actually use MHC, recognizes the antigen or which is present in a cancer cell using MHC. So they are more natural. And coming through other gene therapies, uh, as I told you about beta thalassemia management through Casgevy, and there was a huge success in regaining the eyesight, which is SERP290 associated inherited retinal degeneration, causing several early onset of vision loss. This was ameliorated by a Cas9 mediated gene editing. Then comes the clinical trial of base editing. Base editing was used in CAR7 T cells for relapsed T cell acute lymphoblastic leukemia. And here cytosine deaminase was used. And another good example of gene editing is Cas9 edited CD34 cell, 34 positive hematopoietic stem cell progenitor cells, and the targeted disruption of HBG1 and HBG2, which are the globin gene that promote. So these are the HBG1 and HBG2 are promoters of the fetal hemoglobin. So what is essentially they are doing is that that promoters are very important in gene expression. Here, if you disrupt the promoter, you will not have gene expression. But here, what you want is to increase the gene expression of fetal hemoglobin. So, but you also know that there are elements in the promoter which are where repressors are binding. So repressors, when bind to the genetic elements, the promoter elements represses the gene. So if you remove the repressor sites or disrupt the repressor sites, gene expression will increase. That is another a very clever strategy which was employed by this group in treating thalassemia. Sorry, in treating sickle cell disease. And as I told you already, CRISPR-Cas9 technology was used for hereditary angioedema, a condition where your face or lips or body gets swollen up or inflamed without a clear reason. And CRISPR-Cas9 gene editing was also successfully used in transthyretin amyloidosis. There are different types of amyloidosis. So one type is called transthyretin amyloidosis, where transthyretin, a protein which is synthesized in liver, gets accumulated in different tissues, making that tissue dysfunctional. And there is another recent paper which tells about circularized RNA, which can be used in gene editing, because you always need to provide either the Cas9 protein or the gene editing template. So all this can be given as RNA. But the problem with the RNA is that RNA degrades heavily because body has an immunological response against RNA. So what they have done is that they have circularized the RNA. When RNA is circularized, many RNases cannot act on them. So this improves the gene delivery. Gene delivery or the delivery of genome editors themselves. So as I was telling you, single gene therapy costs millions of dollars. The effect of a gene therapy may be lifelong, or it may save several lives, but it is extremely costly. Luckily, our finance minister Nirmala Sitharaman has announced that gene therapy will be fully exempted from GST. And I will just briefly tell, since I am from a liver institute, I will tell about gene trials, gene therapy trials involving the liver, hemophilia A and B. AV and lentivirus can be used. Then urea cycle disorders. For example, I told you about the death of a boy called Jesse Gelsinger. Here he was suffering from a urea cycle disorder. Then it is useful in Wilson's disease, a genetic disease where copper accumulates in liver and brain. And alpha-1 antitrypsin deficiency I already mentioned. And gene therapy can be of use in hepatocellular carcinoma as well. And what is the future? So far we were editing stem cells, committed stem cells, and all organs or all humans. What about editing or preventing the disease even before birth? That is where we are heading to. We can use gene editing in lab-grown embryos. And quite interestingly, lab-grown embryos increasingly resemble the real thing. As you can see, can you believe that the picture shown here is completely made in the lab? It's a developing human embryo. Three types of cells can create a human embryo: the embryonic stem cells, the trophoblast stem cells, and extraembryonic endoderm stem cells. If you mix all the three types of cells, you can make an embryo, which has the potential to grow into a full baby, perhaps, or a full organism. So, if you can edit these cells, I mean, from pluripotent stem cells, you can make all the three types of cells. Pluripotent stem cells have different states. One state is called a naive pluripotent stem cell. The naive pluripotent stem cells can be converted to embryonic stem cells, trophoblast stem cells, and extraembryonic stem cells. So if you can edit the naive pluripotent stem cells, you can make an entire organism which is gene edited. And that is the future. As you can see, this is the efficiency of development of artificial or synthetic embryos. They are also called synthetic embryos. Or so this is going to be a huge revolution. So summary of the latest developments. We can now edit the genome with huge precision with reduced off-target effects. Off-target effects means reduce the side effects. Gene therapy for sickle cell disease is now a reality in multiple ways, using gene editing or without using gene editing. And now GST second, in India, gene therapy is now exempted from GST, and CAR T cell therapy now heavily used in leukemias, but in future we would be able to use engineered lymphocytes for solid tumors also more effectively. And a gene therapy for heart failure, that is experimental. And future in vivo, we are going to have in vivo gene delivery systems. Probably the development in lipid nanoparticles or completely engineered viral vectors or proteins, maybe using AlphaFold or several artificial intelligence tools, we will be able to develop uh, uh, highly efficient gene delivery tools, which will make the use of virus as a gene delivery tool in future. And editing embryonic stem cells and embryoid models is going to open a completely new area. This is going to make us stronger. It may completely eliminate the possibility of aging, cancer, infections, and a lot. Sky is the limit. Now comes germline editing. If you can edit your germ cells, you probably can have offsprings who are immune to several diseases. Aging is a beautiful target. Senolytic CRISPR-Cas9 therapies or other type of therapies uh, are in vogue, are slowly picking up, which is going to delay aging and frailty. And mainstream applications, expansion into treating diseases like PFIC, cystic fibrosis, Duchenne muscular dystrophy, Huntington disease. These are the mainstream applications. So now I have reached the end of my presentations. Any questions?